The overall goal of this procedure is to provide an automated method to analyze and quantify changes in morphology of an undefined shape cell, taken from three-dimensional confocal fluorescence images. This is accomplished by first performing the chemical stimulation experiment to include cells treated with agonist, cells treated with antagonist followed by agonist, and cells with no treatment. The cells are then prepared for immunofluorescence analysis. The second step is to acquire multispectral three-dimensional fluorescence images. Next, three-dimensional morphometric analysis is performed using Imaris Neuroscience, ImarisXT, and MATLAB computer software. The single-cell phenotypic changes visualized through three-dimensional morphometric analysis are then analyzed and quantified as a final step. Ultimately, this software platform is used to quantify changes in cell shape following receptor activation, providing an additional tool for drug discovery.
Generally, researchers who have a standard expertise in biological systems may not have familiarity to computer application. In this protocol, ImarisXT module bridges the gap between the visualization and computer language.
Prior to the start of this procedure, transfect human embryonic kidney cells with hemagglutinin-tagged corticotropin-releasing factor receptor 2, or CRFR2, as referenced in the written protocol. CRFR2 is a G-protein-coupled receptor, or GPCR. The day after the transfection, flame cover slips and place them into a 6-well plate. Add 10 milliliters of PBS to a vial of 5 milligrams of lyophilized poly-D-lysine and mix. Then, dilute 5 milliliters of dissolved poly-D-lysine in 500 milliliters of PBS, and add 2 milliliters of the mixture onto each of the cover slips.